Hello ladies, welcome to our lecture on meiosis. It's not the same thing as mitosis. It's all about how we make germline cells, or the sperm and the egg. So let's dive in. Before we do anything about meiosis itself, we need to review some of these terms regarding chromosomes because, let's face it, it's a little confusing. So a chromosome, remember, is just a highly coiled or twisted DNA and protein mix. And in eukaryotic cells, it's always found in the nucleus. A chromatid is the name that we give for one copy of a chromosome. Remember, this is a chromatid, and this is a chromatid, and this is a chromatid. And when we have two chromatids like this, connected together, we call them sister chromatids. And they're connected at that centromere, that center point of the chromosome. So now we have to introduce a new term because we're dealing with the copies of our DNA that we get from our mom and our dad. Remember, we have 23 pairs of chromosomes. That means I have chromosome number 1, and a copy from my mom and a copy from my dad. Chromosome 2, a copy from my mom, a copy from my dad. That brings us to a total of 46 chromosomes. So homologous chromosomes are the two copies of the same exact chromosome, like I was just saying, how chromosome 1 has the copy from my mom and my dad. Homo means the same in science, so this is literally saying the same chromosome. And as we learned in mitosis, the sister chromatids are when we make an identical copy of that chromosome. So remember DNA replication, where you have the polymerase and the ligase and the helicase, all that happens during S phase of interphase. A lot of words we're learning. But basically, we start out with one chromatid from our mom and one chromatid from our dad. And let's say this is chromosome number 1, just to make it easy. Well, that's how we start out our cell. Then we go through DNA replication, where we copy our DNA in preparation to divide. That's when my mom's chromosome gets copied and forms that X shape, and my dad's chromosome gets copied and forms that X shape. So we can have sister chromatids, those copies, for both of our homologous chromosomes, the chromosome from my dad and the chromosome from my mom. So in the end, this forms what we call a tetrad. Tetra relating to 4. So it's where both of my homologous chromosomes have copied themselves. So I have 4 chromatids here. That's why it's called a tetrad. That's exactly what's being shown in this picture here. Both of these together, or all 4 of those chromatids together, form the tetrad. And remember, the centromere is the central point where the copies of the chromatids, the sister chromatids, connect to each other. It's just a way for our cells to keep things a little bit more organized so we don't kind of lose track of them and end up moving the wrong chromosomes around. So just to review, this is a chromatid and these are the sister chromatids. However, both of these are going to be homologous chromosomes. Same thing on this side. These are both homologous chromosomes. The copy from, let's say, my mom on this side and my dad on that side, even though, strangely enough, my dad's favorite color is purple and my mom's favorite color is green, so I should have probably done that differently. But in this case, I've copied my mom's chromosome, and in this case, I've copied my dad's chromosome. When we have the pairs from my mom and my dad, it's called the homologous chromosome. And I'm just using my parents as the example. Obviously, we could easily say your parents or your cat's parents or your dog's parents, anything like that. So Remember, every species tends to have its own unique number of chromosomes. Humans have 46, but our chromosomes come in pairs. So we have 23 pairs. So you can see there's number 1 here, 22, and then we have our 23rd down here with our X and our Y. And I have 2 copies of each of them, the one from parent number 1 and the one from parent number 2, your mom or your dad or whoever it is. Dogs have 78. Chimpanzees have 48, which is very similar to ours. And remember, we learned that this picture that I'm showing here is called a karyotype. It's a picture of all of an organism's chromosomes, and we use it to get a really quick snapshot of whether or not they have any inherited genetic diseases. Ones that are really obvious to see, like extra copies of chromosomes or missing chromosomes. We can also use it to really to tell if it's a male or a female karyotype that we're looking at. In this case, I have one X and one Y chromosome. It's always the last pair. So that tells me that I have a male here. Now, if I had 2 X's, that would tell me that I'm looking at a female. So now we are learning 2 brand new terms that I've only sort of hinted at so far. Diploid means when we have 2 copies of every chromosome, just like we've been talking about the pairs, the one from the mom, the one from the dad. Just like this example here, every single chromosome has a pair. We call that 2n. So whatever number of chromosome pairs we have, we multiply it by 2 to get the total number of chromosomes. So humans have 23 pairs, so that would be 2 times 23, and that's how we get our number of 46 total chromosomes. Now, di means 2. Haploid is this new term that we're learning, where it means the organism only has 1 copy of every chromosome. There's definitely some organisms in the world that they live their whole life like this. For example, bacteria. They only have 1 copy of their chromosomes. Usually they only have 1 chromosome on top of that. But you can see on this side, they look very similar, but they're just missing that second copy. So they don't have a copy from their mom and their dad. They'll just have 1 copy of chromosomes. Now, this is what our sex cells or our gametes are called. They're called haploid because they only have half of the genetic information. If this egg has only half of the genetic information, N, and this sperm only has half, it's also N. When we add them together to create a brand new cell, a brand new organism, we get 2n. So it's the egg and the sperm that are haploid coming together to make this 2n diploid organism like us. So this whole process of meiosis is going to be learning about how we make that egg and that sperm. So here are the big ideas before we dive into meiosis itself. The purpose of meiosis is to create our gametes or our sex cells. Another term that's often used for them is germline cells. That's in contrast to the somatic cells that we learned about with mitosis. Somatic cells are the cells of your body that they reproduce through mitosis. The other 2 big ideas are that concept of diploid and haploid. Haploid means we only have 1 copy of every chromosome. Diploid means we have 2. And when the egg and the sperm merge, they create a diploid cell called the zygote. And we're going to go over a little bit more of all this in detail as we learn about meiosis. So what is meiosis? As I said, it's how we make our gametes, our sex cells. We start with 1 diploid cell, 1 normal cell from your body, and by the end, we end up with 4 haploid cells. Essentially, it's very similar to mitosis, but we do it twice. So this picture here, part A, this is showing interphase. We started out with our normal cell, and they copied all of their DNA. We went from one chromatid to a sister chromatid. Then that cell goes through the first half, meiosis 1, which looks very similar to what we just kind of learned about with mitosis, except we're just separating the mom and dad chromosomes from each other. We're not actually pulling the sister chromatids apart. That's what happens in this part here, meiosis 2. So meiosis 1 is going to really only be the new stuff that we learn. Meiosis 2 is going to be identical to mitosis, so it should be kind of easy to remember that. So now meiosis 1, let's get down to it. It's broken down into prophase 1, metaphase 1, anaphase 1, I apologize for that typo there, that should say anaphase 1, and telophase 1. We're going to start out with one diploid cell where there's a tetrad in there, 4 sister chromatids, and we're going to end up with 2 haploid cells. What you're going to end up doing, not right now, but you are going to go back after this PowerPoint and you're going to watch the Amoeba Sisters video on meiosis. It's posted to our topic page, and I'd like you to watch it before our next class. Now, prophase 1, the tetrads are going to form. Just like they did in regular prophase, the tetrads condense, the chromosomes coil up tighter and condense so we can see them. The nucleus is going to disappear, the centrioles and the spindle fibers form, and crossing over occurs. This is the only new thing here. We've never heard of this term crossing over before. Basically, it's where the two chromosomes switch up some of their genetic information. They'll perform a translocation. What we learned about when we were talking about our mutations. So crossing over is when the non-sister chromatids exchange their genetic information. Why do we do this? Well, because it increases genetic diversity. That's a really good thing. Organisms like to be as genetically diverse as possible because this will help us with evolution. It'll help us survive even if our environment around us changes. And we're going to talk about that idea of genetic diversity and how it ties into evolution more in our evolution unit. But here's just a quick teaser. So now, what's going to happen is this leg of one chromatid is going to reach over and cross over with the leg of the second tetrad, or the second sister chromatid, and they're going to switch places. So instead of a tetrad where the two big B's are on one chromosome and the two little B's are on the other chromosome, we've now switched it so that it's going to be big B, little b, big B, little b. And the whole point of this is to increase our genetic diversity. Now, metaphase 1 is similar to regular metaphase, but the tetrads are lining up in the middle of the cell along that imaginary metaphase plate. And the chromosomes are not always going to be on the same side. So I don't have all blue chromosomes on this side and all red chromosomes on this side. No, what happens is they mix it up. Just because I have chromosomes from my mom and my dad doesn't mean that all my moms are going to line up on one side and all my dads are going to line up on the other. That doesn't give us genetic diversity. It actually limits genetic diversity if we do it that way. So we're going to let these chromosomes mix up however they tend to fall. In anaphase 1, the tetrads are going to be separated. So in this case, we're not actually breaking at that centromere. We're not separating the sister chromatids. We're separating the tetrads. And the big thing is they're going to now move to opposite ends of the cell. Those spindle fibers are going to be pulling them to opposite ends of the cell. In telophase 1, we're going to be finishing this all up and we're going to form two haploid cells. The nucleus is going to start to reform around those chromosomes. The centrioles are going to break down, the chromosomes loosen up, and we're going to have cytokinesis occur. So, ask yourself now and quiz yourself, how many cells did we just make at the end of meiosis 1? And were they haploid or diploid at the end? Now, let's go over that second question a little bit here. I'm diploid when I have the copy from my mom and the copy from my dad in the same cell. When we do meiosis 1, I separate those chromosomes. I pull the one chromosome into a new cell and the other chromosome into its own new cell. So I end up with only, let's say, the copy from my dad in this cell or the copy from my mom in this cell. It's no longer diploid. I don't have two copies of the same chromosome. I only have one copy of the chromosome, only my mom's or only my dad's. Now, I know that's kind of confusing because it's still got the X shape. But remember, this is identical information. This happened because I copied my DNA directly. My dad's DNA is always going to be slightly different from the DNA that my mom gave me. So my dad's DNA can have its own copy, but it's still always going to be different than the copy that my mom gave me. So we start out as diploid, or 2N, and at the end of meiosis 1, we end up as haploid, or just N. Now meiosis 2. This is where we're going to separate the sister chromatids. This part is going to happen exactly like it does in mitosis, except it's happening in 2 cells at the same time. At the end, we're going to have 4 unique cells. They're all going to have a different mix of your DNA in these cells. So we're still looking at the same steps: prophase, metaphase, anaphase, telophase, and cytokinesis, and we're separating the sister chromatids this time. We're going to break that X in half and send each part of it to opposite ends of the cell. So in prophase 2, the chromosomes condense. These are now sister chromatids, not tetrads. The nucleus is going to disappear. The spindle fibers and the centrioles form again. In metaphase 2, the sister chromatids line up on the middle of the cell at that metaphase plate. In anaphase 2, the sister chromatids are broken apart and pulled to opposite ends of the cell. So we're starting out with that X, we're breaking it apart, and those pieces are now getting pulled to opposite ends of the cell, and we're going to make a new cell around them. And in telophase 2 and cytokinesis, we remake the nucleus, we make those spindle fibers disappear, cytokinesis is going to happen. And this part's a little bit different for eggs versus sperm, and we're going to go over that. But basically, we're getting 1, 2, 3, 4 unique cells now. So why does meiosis occur? How many cells are formed after meiosis 2? And are the cells haploid or diploid after meiosis 2? Well, at the last point when we checked in, my cells were haploid. They only had that little N. They weren't 2N anymore. And all I did was separate the identical copies of the cell. I still had a chromosome in each cell, so they were still haploid. So we go from haploid to haploid cells in meiosis 2. So what is the difference between mitosis and meiosis? Mitosis is what we call asexual reproduction. I'm taking my copied DNA and I'm separating it to make 2 identical cells. In meiosis, I'm not making 2 cells, I'm making 4, and I go through those phases of separation twice. And in the end, these cells are not identical, and they're going to help me do sexual reproduction. An egg is eventually going to mix with sperm DNA from some other organism. It's 2 different organisms' DNA coming together, mixing to create a whole new unique organism. Now, because we did crossing over, all of my chromosomes mixed up with each other. So now the chromosome that I got from my mom and the chromosome that I got from my dad, they mixed up their DNA a little bit. So now I've got chromosomes with DNA from both of my parents on the same chromosome. So all of this mixing happens randomly. So every single one of these 4 cells is absolutely unique. They're going to have their own mix of all those 23 pairs of chromosomes. So here they can show you a little bit more about the differences between mitosis and meiosis. This one really focuses on the diploid versus haploid. In mitosis, we start as diploid and we end as diploid. In meiosis, we start diploid. After the first stage, our cells become haploid and they stay haploid at the end. You're also going to go home and you're going to watch the Amoeba Sisters video that I've posted on our topic page. This one is comparing mitosis and meiosis. So you're going to really get a deep dive into what's the difference between the two. Now, remember, our big ideas is that the purpose of meiosis is to create those gametes or those sex cells so that we can create a new zygote when the sperm and the egg fuse. Now, I told you that making sperm and making eggs is a little bit different, so we're going to go over that now. Spermatogenesis is meiosis that makes sperm cells. 4 sperm cells are made for every cell that goes through meiosis. Now for men, this starts at puberty and continues until they die. And a fun fact, sperm are the smallest human cells. For women, they go through a process called oogenesis, and that just means making egg cells. And egg cells, fun fact, happen to be the largest human cell. Kind of cool. So this happens very different in males and females. In males, meiosis 1 and 2 happen pretty much back to back with each other. In women, meiosis 1 happens in the womb when you are in your mother's uterus, and then it pauses at birth. Meiosis 2 starts when the egg is fertilized, which is crazy. Now for women, we're born with all the eggs that we're ever going to have. So eventually we're going to run out, and that's usually around the ages of 40 to 60. What we end up making is 1 mature egg and 3 polar bodies. These polar bodies essentially get reabsorbed as nutrients for the big cell, the big egg cell. And when the sperm and the egg fuse, a zygote is formed. We go from that sperm that's N and the egg that's N, and they come together to create a new organism that's 2N. And that cell, that single cell, is going to divide into 2, then that's going to divide into 4 and 8, and it's going to keep going until you're literally a little hollow ball of cells. And then you're going to keep developing until you're a little tube of cells. At 8 weeks, until birth, it's considered a fetus. Anything before 8 weeks, we call it an embryo.